Hello friends, welcome back to a new video, that is MCQs on biotechnology, and this is the part 8 of this series of videos on biotechnology. So the first question of this video, that is 141, question number 141, that is which of the following amino acids is tagged by affinity tag, and the options are option A glycine, option B cysteine, option C proline, option D tyrosine, and the right one is option B, that is cysteine. So cysteine is amino acid which is tagged by affinity tag. So what is affinity tag? So affinity tag are so named because they are often used in affinity purification, a technique for purifying proteins for cell lysis. So in this in this type of purification of protein, the protein with their affinity to other thing or other base, they are purified differently. Okay, so here cysteine have a disulfide bond, so that's why in the affinity tag cysteine is used, cysteine amino acid is used for affinity tag. Okay, so let's move on to next one, that is question number 142. So the small protein present in all cells that play an important role in other proteins destruction are called option A ferritin, option B ubiquitin, option C HSP, or option D degradin, and the right one is option B, that is ubiquitin. So the small protein present in all cells that play an important role in other proteins destruction are called ubiquitin. Okay, so next question देखेंगे. Next है the ability of third base in tRNA can hydrogen bond with any of the bases at the 3' end of codon is or are known as option A mutagens, option B redundant, option C obel, option D tRNA degeneracy, and the right one is option C, that is obel. So this is known as a obel hypothesis. Okay, so what is this? So the ability of third base in tRNA can hydrogen bond with any of the bases at the 3' end of codon is or are obel, and the hypothesis is known as obel hypothesis. So the theory states that the base at the 5' end of the anticodon is not spatially confined as the other two bases, allowing it to form hydrogen bond with any of several bases located at the 3' end of a codon. Like this is a normal pairing where the anticodon here this is the anticodon, this is the codon. Okay, so this pairing is GC right, A and U right, and G and C right. But here you can see this G from anticodon can bind with U. This is not a ideal binding. So this third anticodon can bind any of the any of the bases because their specificity is not mandatory. So that's why it is called the obel pairing. Or and this type of binding is this type of theory. This theory is known as obel theory. Okay. Okay. So let's move on to next one. That is which of the following correctly explains zDNA? And options are option A, this is a left-handed helix of DNA. Option B, it has most base pair per turn. Option C, has alternative purine pyrimidine sequences, and option D, all of the above. And the right one is option D, that is all of the above are correct about the zDNA. That means this zDNA is a left-handed helix of DNA. It has most base pair per turn and has alternative purine pyrimidine sequences in zDNA. Okay, so these are all about zDNA. So next one is the enzymatically inactive precursor of certain proteolytic enzymes is or are option A prosthetic enzyme, option B zymogens, option C ribozymes, option D absence, and the right one is option B, that is zymogen. So the enzymatically inactive precursor of certain proteolytic enzymes is or are zymogens. Okay, so let's move on to question number 146. That is, which of the following plasmid can replicate in both E. coli and yeast cell? And the options are option A YAC, option B BSC, or BAC, that is the yak, that is BAC, and option C YEP, or option D all of the above. So the right one is option C, that is YEP, that is YEP. So which of the following plasmid can replicate in both E. coli and yeast cell? That is YEP or YEP. Okay, so let's see this yeast episomal plasmid, that is YEPs, yeast episomal plasmid are shuttle vectors. They can replicate in E. coli and also in yeast. Okay, so understand. So let's move on to next question, that is 147. So the use of diffraction patterns produced by X-ray scattering from crystals to determine the three-dimensional structure of the molecules is called option A X-ray imaging, option B X-ray crystallography, option C X-ray microimaging, option D none of the above, and the right one is option B, that is X-ray crystallography. So the use of diffraction pattern produced by X-ray scattering from crystal to determine the three-dimensional structure of the molecule is called X-ray crystallography. Okay, so let's move on to question number 148. That is, based on base pairing rule, the bases in double-stranded DNA paired with which are comfortable, or option B complementary, or option C convenient, or option D char graph understanding. So on the basis of which matter the base pairing is done between double stranded DNA. Two strands of DNA can base pair with each other by which of the uh, by depending on the which matter or which base. So the right answer is option B, that is complementary base pairing. So based on base pairing rule, the bases in double stranded DNA pair with which are complementary, okay, which are complementary to each other. So let's move on to question number 149. That is, an RNA molecule binds another one to which it is complementary is called option A universal RNA, option B nonsense RNA, option C RNA world, option D RNA mistake. And the right answer is option B, that is nonsense RNA. So an RNA molecule binds another one to which it is complementary is called nonsense RNA. Okay. So next question देखेंगे. Next is 150. So an ideal hybrid should have option A both nuclear genome and cytoplasmic genome equally from both the parents, or option B nuclear genome from one of the parents and cytoplasmic genome from other parent, or option C nuclear genome predominantly or exclusively from one of the parents and cytoplasmic genome equally from both the parents, or option D nuclear genome equally from both the parents and cytoplasmic genome predominantly or exclusively from one of the parents. So the right answer is option C, that is nuclear genome predominantly or exclusively from one of the parents and cytoplasmic genome equally from both the parents. So that is the ideal hybrid. Okay, so now we should know what is hybrid. A hybrid is a hybrid of cells or plants containing the genome of one species but cytoplasm of both species. A hybrid is also known as a cytoplasmic hybrid. The technique to produce hybrid is known as hybridization. Okay, so let's move on to question number 151, that is advantage of micro projectile method over micro injection method for gene transfer in plant include Option A, intact cells are used. Option B, method is universal in its application irrespective of all shape, size, type and presence or absence of cell wall. Or option C, gene can be transferred to many cells simultaneously. Or option D, all of the above. So the right answer is option D, that is all of the above are correct. So the advantages of microprojectile method over micro-injection method for gene transfer in plants include all option A, option B, option C. These are all advantages of microprojectile. Okay, so let's move on to next question, that is question number 152. That is, what is a usual sequence of Privna box? And options are option A, Tata 80, that is TATA 80, or option B, GATA GATA, or option C, 80 80 80 80, or option D, GC UC GC 80. So the right answer about the Privna box is option A, that is Tata 80. Okay, this is the sequence of Pribnow box. So the next question is, the difference between transfection and transduction is option A. In transfection, the transgene is inserted in a plasmid, while in transduction, the transgene is inserted in a viral genome. Or option B, the transfection involves the transfer of naked DNA into the cell, while transduction involves packaging the DNA into a virus particle which then infect the cell. Or option C, there is no difference, the terms are synonymous. Or option D, term transfection is used for DNA transfer in case of plants and transduction in case of animals. So the right answer is option B, that is transfection involves the transfer of naked DNA into the cell, while transduction involves packaging of DNA into a viral particle which then infects the cell. Okay, so this is the difference between transfection and transduction. So let's move on to question number 154. That is, when present in tissue culture medium, gibberellin helps to break dormancy of buds and bulbs. Or option B, promotes dormancy development in buds and bulbs. Or option C, is degraded as plant growth inhibitor. Or option D, prevents normal recognition of auxin molecules. So the right answer about the gibberellin in tissue culture medium is option A, that is gibberellin helps to break dormancy of buds and bulbs. Okay, so let's move on to question number 155. That is, recombinant live attenuated vaccine against hepatitis B was prepared from option A, plasma of infected individual, option B, recombinant yeast expressing hepatitis B surface antigen, or option C, recombinant vaccinia virus expressing hepatitis B surface antigen, or option D, that is transgenic plants expressing hepatitis B surface antigen. And the right one is option B, that is recombinant yeast expressing hepatitis B surface antigen. So recombinant live attenuated vaccine against hepatitis B was prepared from recombinant yeast expressing hepatitis B surface antigen. Okay, so next question देखेंगे. Next question है 156, that is, which of the following technique is used to inactivate a gene by altering the DNA? And options are option A, homologous recombination, option B, antisense nucleic acid blocks, or option C, antibody microinjection, or option D, introduction of dominant inhibitory mutants. So the right answer is option A, that is homologous recombination. So homologous recombination, which is a technique, is used to inactivate a gene by altering the DNA. So next question is, Question number 157, that is autopolyploidy can be induced artificially by option A chloroform, option B colchicine, or option C chloroquine, or option D colchicine. So the right answer is option B, that is colchicine. Okay, so autopolyploidy can be induced artificially by colchicine. Okay, so autopolyploidy occurs when organisms have more than two sets of chromosomes from the same species. So autopolyploids can originate from issues that arise during mitosis, such as doubling of the chromosomes without cell division. So this would result in a tetraploid organism from a diploid organism. Okay, so autopolyploidy occurs when organisms have more than two sets of chromosomes from the same species. So let's move on to another one, that is androgenic haploid are produced in anther culture by option A tapetum, option B anther wall, option C connectives, option D young pollen grains, and the right one is option D, that is young pollen grains. So androgenic haploids are produced in anther cultures by young pollen grains. So these haploids are called androgenic haploid because they have been developed from male gamete producing spores, that is pollen grains or microspore. So haploid plants originate from gametes or gamete-like cells that do not go through fertilization but can still generate a viable individual. So next one is the efficiency of PCR decreases after a plateau number of cycles because of option A, the already built up PCR product completes with new cycles, or option B, reduced substrate, or option C, scarcity of enzyme, or almost all of the above. So the right answer is option D, that is almost all of the above. So the efficiency of PCR decreases after a plateau number of cycles because of the already build up of PCR product completes with new cycle, reduced substrate, and scarcity of enzymes. So what is plateau? So plateau in PCR can be defined as the attenuation in the rate of the exponential product accumulation, which is seen concomitantly in later PCR cycle. Typically, the reaction will be first exponential, then will enter a quasi-linear phase and finally reach a plateau like this. This is the initial phase, then this is the exponential phase of PCR, and this is the plateau phase. Okay, so this is the plateau of PCR. So the last question of this video, restriction enzyme with a 4 base recognition site would cleave DNA with a statistical frequency of once every option A 256 base pairs, option B 4 base pairs, option C 4096 base pairs, or option D 65.5 kb. So a restriction enzyme with a 4 base recognition site would cleave DNA with a statistical frequency of once every option A, that is every 256 base pairs. Okay, so these are all 20 questions of this part 8 video on biotechnology. Okay, so thank you for watching this video.